Hey guys, welcome to chapter 14.2. In this section, it's a lot shorter, like I said in our Zoom meeting, and we are going to talk about two rules. We're going to talk about the multiplication rule and addition rule as it relates to genetics. So, the probability of tossing a coin, right? When you toss a coin, you can either have a 50-50% chance of getting heads or getting tails. And when you look at Mendel's law of segregation and independent assortment, they kind of relate to that same probability like you're tossing a coin. Because alleles of one gene, they can either segregate into one gamete or into another one. So there's a 50-50% chance of them going into either or gametes, just like tossing a coin. So how do we determine the probability that two or more independent outcomes can occur together in a specific combination? So for example, if I toss a coin, what is the probability that the two coins that I toss in the air at the same time are both going to land on their heads? So this is the probability that we're going to be looking at, right? So if I toss two coins in the air, what is the probability that they are both going to land on their heads? So this is when we use the multiplication rule. We're going to bring in this rule and we're going to see how it works. So what is the multiplication rule? This rule is going to state that the probability of one event, like a coin coming heads up, and you're going to multiply that by the probability of the other event, which is the other coin coming heads up, all right? So multiplication rule is to determine the probability. We have to multiply the probability of one event, like one coin coming heads up, times the other event, which is the other coin coming heads up. So remember how I said if you toss a coin, the chance that that coin is going to come heads up is 50-50, which is 1/2. You're going to multiply that with the chance that the other coin is going to come up heads, which is 50-50, which is 1/2. And if you multiply those together, remember you multiply across and you get 1/4. Now 1/4 is a probability that the coin will come up heads up. So this is how basically you do the multiplication rule. So when we talk about segregation of heterozygous plants, it's pretty much like flipping a coin. Each gamete is going to have 50-50 or half a chance of carrying the dominant allele and half a chance of carrying a recessive allele. So remember, when we talk about a heterozygous plant, right, we're talking about, for example, if I say capital P, little p, that's heterozygous for purple. So the big P and the little p, this P has a 50-50% chance of carrying of going into a gamete, and then this has a 50-50% chance of going into either or gamete. So I give you an example here. If I have capital R, little r, and then capital R, little r, the capital R is going to have half a chance of getting passed down into one gamete, and then the chances that the little r will get passed down is also half, which is 50-50. Same thing on this side. The chances that this big R is going to get passed into one gamete is 50-50 versus the other, and the chances that the little r is going to get passed into another gamete is also 50-50. And so that's how you break it down. So if I have, for example, this allele or traits from the egg, and then I have this one from the sperm, let's break it down. So the chances that the big R is going to go into one gamete is half. The chances that the little r is going to go into one gamete is also half. Chances that this big R is going to go into a gamete is 1/2 or 50%. And same thing with the little. So when I cross it, it's going to look like this. Oops. okay? And so when I cross it, it's going to look like that. And so I can say that this is 1/4, this is 1/4, this is 1/4, and this is 1/4, all right? So you can see just a variation in all of. So now you can see that the probability that we're going to get capital R is 1/4. Probability of getting capital R, little r is 1/4. And so the way that we can break this down is that, so the probability of this is going to be 1/4. And if you look, the probability of this is 1/4. Okay, so now we can further simplify this and say that these two are pretty much the same thing, right? They're both capital R, little r. Even though they're coming from different gametes, one is, the blue is from the sperm and the pink is from the egg, we can still simplify that and say that RR, the probability of getting big R, little r is 1/2, okay? So to further simplify, you can say RR probability would be 1/4, big R, little r is 1/2, and then little r is 1/4. Okay, so now let's talk about the addition rule. We just talked about the multiplication rule. Now let's talk about the addition rule. Notice that when you have a sperm, and it has, for example, say it has RR, it can only give one of these, right? Only one of these can be passed down. It can be either the big R or it can be the little r, but it can't give both of them because remember, one comes from the mom and one comes from the dad, right? So same thing with the egg. If I have, so let me color code this for you. So if I have a sperm that has RR and I have an egg that also has RR, they can only give me one allele from each, the sperm and the egg. They can't give me both of them. So in the multiplication rule, we saw that the individual probability and how it was occurring. But in the addition rule, we're going to see how it works when we add those together. Okay, so we can use the addition rule to figure out the probability of the F2 plant from a monohybrid cross, and we will be able to see that it's going to be heterozygous rather than homozygous. Okay, so we can use the addition rule to figure out the probability of the F2 plant or F2 generation from a monohybrid cross, and we can see that it's going to be heterozygous rather than homozygous. So for example, if I say the probability of one possible way to find this out is find out how we can get a heterozygous F2 is by having a dominant allele from the egg and a recessive allele from the sperm. And the probability of that would be 1/4. So remember in the previous slide how I did the Punnett square and we came out with what it looks like over here. And I said that the probability that a egg, so if I did over here, so this was our male, this was our female. So the probability that the egg, which is right here, is going to give us a dominant allele, which is this R, and the sperm, which is over here, is going to give us a recessive allele, which is this little r, right? That would look like this. So which one of the squares in the Punnett square looks like this? It's this one right here, right? So what is that probability? It's 1/4. So basically it really helps to remember that, make sure that your sperm is here and your egg is over here. And that way, if you want to break it down by colors as you're initially doing it, so you can see how the addition rule actually works, it will make more sense to you to break it down, see the probability of that individual. Now, the second possibility is that if the egg, right, so if I had the egg and I had the sperm, what if they flopped position? So what if the sperm gave the dominant and the egg gave the recessive? So that would look like this. So which one in the Punnett square looks like that? It's this one, right? And so the probability of that is 1/4. So the probability that you would have this dominant from the egg, recessive from the mom, or from dominant from the sperm and recessive from the egg, is 1/4. So when you use the addition rule, we can calculate the probability of the F2 heterozygous, and we can say 1/4 plus the 1/4. So 1/4 dominant recessive, dominant from dad, recessive from mom, or dominant from sperm, recessive from egg, plus the recessive from sperm and dominant from egg will give you 1/2. So the probability that it's going to be F2 heterozygous is 1/2. Okay, so one possible way that we can get a heterozygous F2 is by having a dominant allele from the egg and a recessive allele from the sperm. So if I show you how this looks, let me give you, let me write down the alleles from the sperm. And then the allele from the egg that it could possibly give us, okay? Now, when I have this, like I said before, the sperm can only give me one of the two. So it can only give me a big R or a little r. The egg can only give me a big R or the little r, right? But in this case, one possible way that I can have a heterozygous F2 is if the dominant allele comes from the egg, and if the recessive allele comes from the sperm. So when I combine these together, it's going to look like this. Now, if I look at my Punnett square, this is the same Punnett square that I did in a couple slides ago, and we broke it down. Which one of these in the squares of the Punnett square looks like what I just got, the big R and little r? It's the one that I have circled in yellow, right? So this one, big little r, is from the sperm, and big R is from the egg, and that's 1/4, right? And so I know you might think, oh, but Mrs. Fry, there's another 1/4 that has a little r and big R. But remember, this is being specific to where each R is coming from. The other one right here, the one that I'm circling in white, it has little r from the egg and big R from the sperm, but that's not what we're looking for. We're looking for the dominant allele from the egg and recessive allele from the sperm for this possibility. So according to this possibility, this is going to give us a 1/4 probability that this is going to be the outcome. A second possibility is a recessive allele from the egg and a dominant allele from the sperm. So if you look at the Punnett square, which shows you the recessive allele from the egg. Remember, the egg I put in pink. And then the dominant allele from the sperm would be capital R. So you can see clearly, I kind of gave it away. It's the one that I have circled in yellow. And the probability of that happening is 1/4. So with the addition rule, what we can do is to calculate the probability of this F2 heterozygous is to add both of those probability. The probability from possibility number 1, probability from possibility number 2, and when you add them together, you get 1/2. So the probability that the F2 will be heterozygous is 1/2, because you're taking the probability that each sperm and egg can give to cause it to be heterozygous. Because if you remember, let me show you here. In this one, you can see that the other options, which are big R, little r, they're homozygous, right? But we're not looking for homozygous, we're looking for heterozygous. And so the only way that we can get heterozygous is if it's big R and little r. And we have to break it down into which gamete. Is it the sperm that's giving the big R or the little r, or is it the egg that's giving the big R and the little r? And you have to multiply, you have to add those two probabilities together. Okay, so now let's move on to multiple crosses. The thing with multiple crosses is that we can solve really complex genetic problems with the rules of probability that we just talked about. So we can apply the rules of probability to predict the outcomes of the crosses that have multiple characters, right? So in the one that we just did an example of, we used big R, little r, which could mean round, right? But we could have to do genetic crosses that have multiple characters, like they're round and wrinkled, or they're round and yellow, or they're green and wrinkled. And so what basically multiple crosses means is it is equivalent to 2 or more independent monohybrid crosses occurring at the same time. So even though we might have yellow with round, you have to break them apart to look like as if they were individual, even though they're occurring at the same time. So how do we calculate this? To calculate the different genotypes, like I said, the yellow with the round, you have to take each character, like the color and the shape, and you have to consider them separately. So you're kind of having to split them apart. And once you split them apart, you multiply the individual probabilities together. So one way that we can kind of split this up is by using a Punnett square. So if we're trying to figure out what's the probability that, the offspring are going to have which allele, you would pretty much break them apart into the Punnett square. So usually a lot of people who are really quick at this, they can do kind of like a cross, kind of like foiling. So I'll show you that really quick. So watch me as I draw the arrow from the Y to the R, and then watch me as I write it down what they would be. So for example, if I take the first Y with the first R, I'm going to get this. If I take the same first Y with the second R, I'm going to get this. Okay, so you probably see the pattern now. Now I'm going to do it again, but with the second Y and the first R, and the second Y with the second R. So you can see that they're all the same, right? So you're going to get 100% yellow and round. But let's look at something a little bit more complicated and see what happens. So if I have big Y, big R, little r, I'm still going to have dominant for yellow, dominant for round. But the way that I'm going to kind of split apart this multi-character genotype and see what the probability for each one is by splitting up the Y's and the R's from each other, figuring out their individual probability, because remember how I said in this slide with the multiple crosses that I have to consider them separately and multiply their individual probabilities. So that means I have to figure out the probabilities for the Y's and probabilities for the R's. And the way that I'm going to do that is by pretty much doing what I did over here, which is I took the R's and I split them apart because I'm saying that each R has a probability of 50-50 from the egg or the sperm. So we're going to do the same thing here. So if I take a Punnett square, I'm going to say that big Y, little y on the top, and each one of those y's has a 0.5% chance of coming from either gamete, and same thing on the side. And now I'm going to cross them, and we're going to see what their probabilities are going to be for each type. So let's break them apart. Big Y is going to have the probability of being that, which is 1/4. Big Y, little y, because there's 2 of them, I'm going to say 1/2. And then little y is also going to give me 1/4. Now let's do the same for the R's. I'm going to do it over here. I'm going to split the R's, and I'm going to say big R, little r, big R, little r, and then I'm going to cross them. And the probability would be that big R is 1/4, big R, little r is 1/2, little r is 1/4. I can say that what would the probability be if I had 2 of these genotypes? So if I have the genotype big R, big Y, or big Y, big R, which means it's yellow and round, what would be the probability of this genotype, right? So I can say, well, big Y is 1/4. I'm going to multiply that with big R, which over here is also 1/4. If I multiply that across and from the bottom, I'm going to get 1/16. So that's the probability of having this genotype, okay? Now, if I switched it and I said, what is the probability of this genotype, which is big Y, big R, I can say, well, if I look at my Punnett square with my Y's, big Y, little y is 1/2, right here. And big R is 1/4 when I look at my Punnett square with the R's. Okay? If I multiply that across, I'm going to get 1/8. And there you have the probability of big Y, big R. So the main thing that you have to remember with multiple crosses is even though it's a little bit more complicated, you have to really split them apart, see what the probabilities of them are individually before you multiply them together when they ask you what a specific probability is of a genotype. So if they ask me what the specific probability of the genotype is for big Y, big R, and big Y, little y, big R, I'm able to calculate it easily because I can just look at my 2 separate Punnett squares and multiply the probability that I have for each individual character, right? Character for color and shape, and therefore I can get them together. And that would be it for this section. I know it's shorter, but it is a lot to take in, so please make sure you really understand this concept. If you have any questions, email me.